we look at this part of the plankton called pteropods, and they're holoplanktonic gastropods, meaning that their snails which spend their whole lives in the water column. So in this video, you can see some pteropods. They are the uncoiled sea butterflies, some coiled sea butterflies, and sea angel that's flapping around. So pteropod means wing foot, and they use their wings to navigate along with the water column. And the sea butterflies are important components of the marine food web because they're eaten by sea angels, fish, and even whales. They also have thin, transparent shells made of aragonite, and this means that they contribute to carbon flux in the ocean. Pteropods are known as bioindicators of ocean acidification because of their thin shells. Here we see a video of micro-CT scans of a pteropod. And we know that if we see changes in their shell thickness or signs of dissolution, they can be used as an early warning system for detecting changes in ocean acidity. We also think that different species may have different responses to ocean acidification. Therefore, it's important to be clear about their species boundaries when using them as bioindicators. We focus on this species of shelled pteropod, Limacina bullimoides. We know that they hatch from eggs into veliger larvae and then mature first as males before becoming female later in life. They also have internal fertilization and lay egg strings, as indicated by the arrow over here. Limacina bullimoides is found in tropical and subtropical oceans of the world, and we want to find out if they are one circumglobal species or if they are composed of hidden diversity. We can also understand the extent of local adaptation by looking at the spatial distribution of genetic variation. So for example, we use two barcoding genes to find that in the Atlantic Ocean, this species is actually composed of 3 different populations. Besides this, we can also look at the amount of standing genetic variation within a population to assess if they have enough genetic resources to cope with future changes. Therefore, by looking at the level of genetic diversity both within a species and within a population, we can have a better understanding of their adaptive potential. Furthermore, if we want to have an idea of their evolutionary history, we can look at their genome-wide data. Because of their small size and large genome, as well as the need to sequence many individuals to study their population structure, we decided on this target capture approach where we looked at around 2,900 regions across the genome. This was done by using a draft transcriptome mapped to the draft genome to obtain these regions, such as biomineralization candidates, protein coding genes, non-coding sequences, and some barcoding regions, and these regions were amplified before sequencing. So we had a global data set of 161 individuals of Limacina bullimoides spread across 4 ocean basins, such as the Atlantic, Indian, North and South Pacific. We then did a PCA plot as shown over here and found that there are 3 distinct genetic lineages. There's one lineage that is composed of only Atlantic individuals, another lineage that is composed of Indian and North Pacific specimens, and the third lineage has North and South Pacific specimens. This is also confirmed in the admixture plot, where 3 was the best number of ancestral populations. We can also see from both of these plots that there is no sign of recent admixture or hybridization because these clusters are pretty much distinct with no intermediates, and also the probability of assignment to an ancestral population is 100% for each individual. For the rest of the talk, I'll be mentioning these 3 lineages, such as the Atlantic in blue, Indo-Pacific in green, and Pacific in orange. We looked at heterozygosity as a measure of genetic diversity and found that the Pacific lineage had the most diversity, while Atlantic had the least. We also looked at global weighted FST, which is a measure of subdivision within a population, and found that the Atlantic had the highest amount of substructure despite its lowest diversity. We then did a co-ancestry matrix, which is a pairwise comparison of how related one individual is to all other individuals in this data set. So blue indicates a high amount of relatedness, where yellow indicates a low relatedness. So if we focus on the dark squares over here, we find that these correspond to the same genetic lineages, Atlantic, Indo-Pacific, and Pacific, and there is no mixture between lineages. We also find evidence of geographical substructuring. For example, in the Atlantic, it can be further divided into North, South, and equatorial populations. And this was also seen in our previous study of the Atlantic population where we used barcoding genes. Within the Indo-Pacific lineage, the Indian Ocean individuals were more closely related to each other than to the rest of the individuals. As for the Pacific lineage, North Pacific individuals were more closely related to each other. We also used a SNP phylogeny with around 1,000 SNPs to get a better idea of their evolutionary history. This tree was calibrated according to the time of divergence from its sister species, Trochiformis, at 13 million years. We included 2 specimens of the outgroup Trochiformis and 10 individuals each from the genetic lineages. And we found that the 3 lineages diverged at around 1 million years ago. From this method, we can also estimate the effective population size, which is the highest in the Pacific at 6 million, followed by the Atlantic at 3 million, and Indo-Pacific at 1.6 million. And these are pretty high numbers. And then we looked at the shell shape. So we placed landmarks as indicated by the yellow dots on the shell margin and shell aperture. And then we did this for 159 individuals for which we had genetic data. Then we did a relative warp analysis, which is kind of analogous to a PCA. And this is what we find. So these are the axes of the most variance, relative warp 1, explaining 51% of the shell-shaped data, and relative warp 2, explaining 18% of the shell-shaped data. Each dot is a specimen, colored according to their lineage, and we have some representatives of the specimens shown over here. And these plots over here show the shell-shaped deformation, and the dark dots corresponds to the landmark. So if we look at these 2 over here, we find that relative warp 1 is correlated with changes in shell width, while relative warp 2 is correlated with changes in the shell aperture. We can see from this plot that there's quite a bit of overlap in shell shape between lineages. Therefore, we did another analysis, which is a canonical variate analysis. And what this analysis does is that it maximizes the differences between lineages while minimizing differences within lineages, so there's better separation of the groups. And then the colored regions represent the distribution for each lineage, while the dark circles represent the 95% confidence interval. And then with this plot, we can see that the mean shell shape is significantly different for each lineage. However, there's also a large amount of overlap between lineages. Therefore, we can't really assign specimens to their lineage based on their shell shape. So this is a map of the genetic lineage assignment for each location as pie charts. If we focus in on the tropical Pacific, we find that there are interesting things happening over here. So near the Hawaiian Islands, there are 3 locations where we find 2 lineages, Indo-Pacific and Pacific, at the same spot. This means that they're reproductively isolated lineages since they occur together. We find that despite the different genetic backgrounds for these 2 lineages, the shell shape is not significantly different, so we can't tell them apart based on their shape at all. We then decided to look at other aspects of their shell morphology because there must be something we can find. So this is a bit of a challenge to everyone. You can try to see if you can spot the difference between the two lineages, and I'll just wait a bit while you guys stare at it. Okay, so I'll just say the answer. And if we zoom in closer, we can see that actually the Pacific lineages, they have some pigmented spots on their tissue that is not found in the North Pacific. So this might be something worth looking into. So to conclude, Limacina boulemoides is not a circumglobal species. Rather, they're distributed into 3 separately diverging lineages, and there is no evidence of recent admixture or hybridization between them. This means that they're reproductively isolated, which is also confirmed by the presence of sympatric lineages in the North Pacific. For further work, I would like to study functional variants in the Atlantic to get an idea of local adaptation. And then we find that these lineages have been evolving independently since about 1 million years ago. So it's pretty likely that their genomes are different and they will have different responses to future stresses. This, coupled with their high standing genetic variation and large effective population sizes, means that they may be more resilient than we think to future ocean changes. Thank you.